That's actually something I've not talked about much on the podcast. And so I'm wondering if you tell me a little bit about what the basic setup is in your lab for studying things like alcohol consumption and compulsive alcohol taking in rodents and what that looks like sort of in comparison to humans.
So rodents are actually an excellent model. They will drink, as you mentioned. So I'm familiar with Christian Lucher's work. They drugs like cocaine and heroin are readily adaptive for a lot of rodents. Alcohol is less so. They do have a very protective mechanism where if something tastes different, they'll avoid it. It's called
Mm-hmm.
neophobia. So it is not that they go binge immediately voluntarily. You do have to get them to become used to it, understand that it's not going to kill them immediately, and then they'll drink. So we have studies where they will voluntarily drink in their home cage. We have studies where we experiment or administer alcohol, and we can do that through intragastric means, through an oral gavage. We also have vapor inhalation. So they'll sit in chambers and just breathe in alcohol vapor and get to very high BALs in a very relatively stress-free environment. But in all those cases, there's pluses and minuses. So with the home cage drinking, they have the option of choice and volition, which are very essential and
Mm-hmm.
most relevant to human behavior.
And
Um,
what literally are they drinking? Is it like a 10% ethanol solution? Is it
so we use a 20% ethanol solution, and this is 20% lab grade ethanol. So no flavors, no liquor types. It's
mhm.
just the 20% ethanol that tastes like ethanol. And they'll still drink it. So that I think is interesting because certainly it's not super palatable. And there is, I think, some relevance to be made for some of the drinking that we likely see in the lab setting is because they're bored
Mhm. Yeah,
and
yeah.
with something new. I don't think that that doesn't relate to the human condition entirely, but importantly, yeah, they will consume alcohol. And interestingly, though, there are strain differences. So we often will say rats and mice together, but it's been well known in the field for a long time. Mice will drink quite a bit, and it is strain specific. So we work with a strain called C57 Black 6J. It's a lab strain that's been used for a very long time. They consume very, very high quantities of alcohol voluntarily, and there's a sex difference there.
Hmm.
The females will consume considerably more than males.
Females
By,
consume more than males.
Mhm.
I would guess that it's the opposite in human beings, but I don't actually know for sure.
Well, so this is where humans are a little more problematic than rodents.
Yeah.
Rodents don't have any cultural bias.
Hmm.
They don't have any like cultural expectations for alcohol or
Yeah.
what it might be to them. This is just pure male and female. And the females, yeah, they drink more. They do also, in a lot of instances, drink more water. So there could be drives for fluid intake that come into play for females that aren't as relevant in males.
Hmm.
Um,
And
but
so,
all of those are interesting.
yeah, and can you give us a sense for in human beings, like how do psychologists or psychiatrists define alcohol use disorder? What does that look like in terms of how much people drink before it's clinically considered to be a problem? And how do you then start to model that in the rodents?
Well, so this is a really good question, and this is something where a lot of progress has been made in the field, appreciating the complexity of alcohol use disorder, because historically, people would use very stigmatized language like alcoholic, drunk. This really was a problem clinically because it kind of suggested that these people had problems controlling themselves. They had issues of, discipline and self-control and moral failings. Um, that's all a cultural baggage that humans have, and we sort of progress from there, based in large part by preclinical work. Rodents have none of this. They have no motivation for any of this other than what they can choose to do on their own, and they will drink. And you can see in rodents, as well as other models like non-human primates, they will regularly consume and they'll achieve some of the same things that are now in the diagnostic criteria for human alcohol use disorder. So that would be escalated intake, that would be somatic withdrawal signs in the absence of the drug, that would be increased seeking and taking, escalation, the development of tolerance. It's a very multifaceted disorder, and that's why I think currently it's not a yes or no question. It's of these range of criteria, how many resonate with you, and past a certain limit, you are now qualified for an alcohol use disorder diagnosis.
Mm-hmm.
So it's not one thing, it's sort of a spectrum, a constellation of effects.
Mm-hmm. But so it's a spectrum of effects. I think the two key ingredients here, it sounds like, are one, dependence or tolerance. So you develop a dependency such that in the absence of the substance, you feel bad. There's measurable things happening, and the other would be like compulsive seeking, like giving up doing certain things that would be considered ordinary in order to acquire the substance.
Yeah, I think the term compulsivity is really controversial in the field right now because that is harder to model in animals. But they will say more like habitual, so repeated, consistent intake. And this is something we can absolutely see in our models, the voluntary models. You give these animals access. So when I mentioned home cage drinking, they're not forced to drink. There's a bottle of alcohol and there's a bottle. They can choose to leave the alcohol entirely alone, and some animals do.
Mm-hmm.
So going back to the differences you mentioned, it's absolutely the case that there are some that will sample it and then decide it's not for them and choose to consume almost nothing or very low levels forever. There are some that will sample and increase their intake over time. There are some that will start high and stay high. I mean, how it looks, even in a reduced rodent model, is as variable as it is for humans.
Interesting. Yeah, I was going to ask that next, actually, the individual variability piece. So there's quite a bit of variability individually, also strain by strain, species by species. So all of the above.
Well, and this is where you get at some really interesting things, because going back to some of these psychiatric conditions, an area of intense study is the overlap and comorbidity and relationship between things like anxiety and depression and substance use. And that shifts your landscape, because if you're an individual that has a pre-existing anxiety-like disorder, you may consume alcohol for partial relief of that anxiety. And absolutely, that's something that's been reported clearly with social anxiety. Alcohol is really well known for being what's called a social lubricant. And this would help somebody that is trying to relieve their negative feelings.
Yeah. And I guess I'm just going off the cuff here, but I think you said mice tend to drink more voluntarily than rats do. Yeah. And if you think about this, like in ecologists, right? These are both prey species, but rats will prey on mice, and mice are smaller and more vulnerable. So you might expect them to have higher baseline anxiety, and maybe that is potentially a tie-in here.
Yeah, we don't. So this is actually where it's in our lab, we look in rats and mice, and we perform a lot of anxiety-like or depressive-like tests in rats and mice. And I haven't seen what you're describing, this idea of these mice being more anxious and that might drive their intake. I'm not sure that we can reliably make that conclusion. And both rats and mice, if you stress them out, you can induce a stress-like relapse phenomenon in both rats and mice. So I think I'm not sure it's as ecologically simple as lower order, higher order. Actually, this is where, going back to the complexity, I forgot to mention, so that C57 Black 6J strain that we used was sequenced, and there's a genetic polymorphism in those animals, in the GABA A receptor, that drives their intake. Other strains of mice drink considerably less and do not have this polymorphism.
What is the effect of the polymorphism in terms of so it's GABA A receptor? We're talking about inhibitory neurotransmission. Are they? Is there less baseline GABA activity in the strain, or more? What's the actual effect there?
No, so it's a subunit of the GABA A receptor, so it's not as easy as less or more. It's receptor distribution, receptor function, which changes by brain area.
I see.
So
but ethanol directly acts on GABA receptors. Is that true?
Yeah, the GABA A receptor is one of the primary, probably the hallmark primary target of alcohol. And alcohol will generally exacerbate the effects of GABA A receptor, which the GABA A receptor is the primary inhibitory amino acid transmitter. So if you think about that inhibition, a lot of the effects we see with alcohol, sedation, slowing of locomotor responses, eventual loss of consciousness, a lot of those are very much under the purview of the GABA A receptor.
One last little question here. So some mice, some rats will voluntarily ingest some amount of alcohol depending on the individual. Do they get drunk? Do they stumble around?
Well, so yes and no. You absolutely can see motor impairments. There's something called a loss of righting reflex. So mice, being prey animals, are not like puppy dogs. They don't like to be turned on their back and have their bellies exposed because that's a very vulnerable, unsafe position for them. So it's an adaptive response to flip yourself back over as quickly as you can. And when animals have a certain level of intoxication, they lose that ability to right themselves, or they will right themselves much slower.
Mm-hmm. And so what are some of the things that you guys have found in terms of the neural basis of the ethanol intake and the development of high levels of ethanol consumption in rodents? Are there particular brain regions that are involved in this?
Well, so this is a perfect circle to where we started with the psychedelic work. A region that is also a central anatomical target of alcohol effects is the central amygdala. And that, again, goes back to the good and bad of the central amygdala. Canonically, the central amygdala has been sort of ascribed to this negative affect that emerges following chronic alcohol exposure. But you can even see that with initial alcohol exposure, the central amygdala is engaged. So that job of assigning valence is happening from the first drink you take, and changes in the activity of the central amygdala will change your drinking behavior. And that is something where I think, just to tie everything together, one of the components of the central amygdala that's very sensitive to alcohol and changes with acute and chronic alcohol exposure is the corticotropin releasing factor receptor 1 system, which is the primary receptor system for the primary stress peptide corticotropin releasing factor, or CRF. But this is also a case where there was a lot of excitement about the really, really beautiful definitive preclinical data looking at the CRF1 receptor. They put this into clinical trials, and they used CRF1 antagonists in human alcohol use disorder patients, and they found relief of some of the anxiety-related behavior, but there was not substantial reductions in voluntary drinking. So this was something that had a strong preclinical backing. It looked beautiful. You could give a CRF1 antagonist and you can significantly reduce voluntary drinking in rodents. It can happen in humans.